Hi everyone, welcome back to Lonza's podcast on the latest trends in pharma manufacturing. Today we will explore the intricate world of producing and utilizing antibodies as therapies. In this episode, we are taking a close look at monoclonal antibodies and even more complex antibodies, and how they are being employed in healthcare. And while it has been more than 3 decades since the first monoclonal antibody was approved by the FDA, today, major advancements in biomedical research and the rapid translation of basic research into clinical practice have expanded the monoclonal antibody industry exponentially. They now represent the fastest growing group of biologic therapies.
What really excites me and what I'm seeing more and more in the industry being reported on is around precision medicine, more targeted therapies, and improvements in delivery systems.
But what does the future hold for these life-changing therapies? You'll hear more about that shortly, including how they are developed, manufactured, and continually innovated. You're listening to A View On, a podcast brought to you by Lonza. To explore the exciting world of monoclonal antibodies, bispecific antibodies, and even more complex proteins, I am joined today by 2 experts in the field. Ronnie Wei, head of Biologics Discovery and Development at ModeX Therapeutics, who's responsible for developing novel, multi-specific therapeutics for immuno-oncology and infectious disease indications. And also Atul Mohindra, who is the head of R&D for biologics within Lonza. All right, so let's begin then. I would first like to thank you, Atul, and you, Ronnie, for joining me today on the podcast. It's a fantastic topic. I'm so excited to talk to you today.
My pleasure. Thank you for the invite, Martina.
Yeah, same here. I'm really excited to discuss with you two on one of my favorite topics, antibodies.
Ronnie, would you mind explaining to us and the listeners what function do antibodies serve in our bodies, and how are they made in our organisms?
Sure. So antibodies are specialized Y-shaped proteins. They are part of the amazing immune defense mechanism our bodies utilize. They are made of a heavy chain and a light chain, 2 copies of each. Antibodies actually are quite versatile. So first, they can identify and bind to specific parts of pathogens, or what we normally call antigens, with really exquisite specificity and affinity. And once bound using the 2 arms of the Y-shaped protein, antibodies can directly interfere with a pathogen's ability to infect cells or function properly, essentially neutralizing them. Antibody can also serve as signposts to call upon other immune cells to either engulf pathogens or other immune proteins. These proteins can punch holes in the membranes of these bugs, effectively killing them. Remarkably, antibody-producing B cells can turn into memory B cells that can be reactivated to produce really large quantity of the protective antibodies very quickly upon re-encountering the same antigen. This way, they also reduce the risks for reinfection from these pathogens. This is actually the basis of modern vaccination.
That sounds fantastic. And it's, I think, a topic that since 2020, the entire world has become quite familiar with as a concept, right? So that all sounds great, but antibodies are then produced by our own immune cells. How can they be used as therapies then?
With the advancement of modern recombinant DNA, antibody engineering, and cell culture technologies, antibodies really have transformed medicine by offering a very targeted approach to treat multiple diseases. To give you a few examples, first and foremost, they are used to prevent or treat infections because of their neutralizing activities. And recently, like you mentioned, since 2020, monoclonal antibodies or mAbs are in general, lab isolated and produce clones of highly specific antibodies that can also be designed to target and inactivate or destroy human cells or proteins that drive pathology. For example, cancer cells, autoimmune, or inflammation driver cells or molecules.
What we've discussed so far are all monoclonal antibodies. We are increasingly seeing more complex antibodies in the pipeline, though. What's your take on this, Atul?
I think the current format is the monoclonal antibodies that we're seeing, which are more traditional in terms of their structure. What we are seeing more in the pipeline, but also in the industry, is new molecular formats, so bispecifics, trispecifics as examples. And they're really designed specifically and engineered specifically for targeted diseases that they're trying to go for.
Ronnie, your company's focus on these new molecular formats, including more complex antibodies that can bind to multiple antigens at once, so trispecific or even multispecific antibodies. What can you tell us about them, and what are they used for?
Yeah, this is actually what I'm most excited about. So there are several advantages of simultaneous targeting multiple antigens or multiple targets. In the most straightforward fashion, multispecific antibodies can prevent antigen escape or overcome disease heterogeneity, often associated with infection or cancer, in a manner similar to using antibody cocktails.
Could you explain what an antibody cocktail is? I mean, does it mean using multiple monoclonal antibodies, so MAPs, at once?
By antibody cocktails, we mean combination of multiple MAPs in the same treatment, but with multi-specific antibodies, we can achieve the same purpose with just one molecule. Also, if we choose the antigens carefully and design the right format, we can potentially reach greater efficacy with a multi-specific antibody over mere combination of the parental MAPs through synergistic binding. Last but not least, multi-specific antibodies can also achieve greater efficacy through forming what we call obligate interactions. This is not possible with MAP combinations. It's a really remarkable advance in recent years. With the combination of antibody engineering and also our understanding of antibody structures as well.
How do the platforms and technologies that your company is developing come into play here?
Yeah, so at ModeX, we actually quite routinely produce tri, or even tetraspecific antibodies. So we choose the targets very carefully and design the format, be it multi-specific antibodies using a more traditional format or sometimes antibody fusions. Really is trying to serve the desired biological functions or outcomes for specific disease indications. So, of course, the challenge is to balance the breadth, specificity, and the potential toxicity to achieve the most desirable therapeutic outcome.
Great stuff that Ronnie and Atul have shared about what monoclonal antibodies are and how they are used in our bodies or in healthcare. We also discussed more complex molecular formats that are on the rise, bispecific or even multispecific antibodies, which can bind to multiple antigens at once and thereby bring greater efficacy in therapeutic effects compared to standard MAPs. Now I would like to focus also on the manufacturing side, right? So one thing is, of course, designing drug candidates, but eventually if we want to get these products to patients, we need to ensure that they can be manufactured and that they can be manufactured at scale. So Atul, a question to you. In general, how does one manufacture antibodies in the first place?
Great question, Martina. So typically the first step in the process is what we would term in the industry the lead candidate selection stage. And what we're really trying to achieve there with the help of our customers is to identify the best sequence possible as they are discovering their lead candidates. So we typically perform a number of screening exercises and use our prediction tools, such as the immunogenicity and manufacturability tools, that would help to guide what is a lead candidate for success. This then feeds into a development program, and here what we would typically do is to express the gene of interest into a host cell expression system using a plasmid vehicle to find the most productive and stable clone. We would then typically amplify that clone and evaluate the performance in a cell culture system. And then we would purify that material and use some of that material for formulation development, because once we produce the drug substance, we would then want to produce some drug product material for which we need to assess the best formulation for. And we would use the rest of that material for other assay development activities, for example. What we would then do is to test the scale-up. And what we would do is typically use that same clone and process that we've just developed in small scale and scale that up to provide material for tox supply or first-in-human clinical supply. Once we've typically provided the phase 1 material, the clients would perform the phase 1, phase 2 studies, and we would then start eventually during the product life cycle, start scaling that up. And of course, at Lonza, we have the scale-up capabilities to go from 1,000-liter scale all the way up to 20,000-liter scale.
Ronnie, what's your take on the manufacturing needs of antibody-based products as someone who works in the discovery stage?
I also want to actually echo what Atul said, the importance to ensure that these antibodies are manufacturable. So this has become a really important emphasis in even our discovery process. We want to make sure the antibodies, and especially the multispecific antibodies we design, are manufacturable and or developable. Thank you, Atul, for all of your hard work downstream to make sure we can put these amazing molecules into patients.
No problem.
You've mentioned a couple of expression systems that could be used in manufacturing. Could you run us through what's available at Lonza and beyond? I'm also particularly curious about plant cells.
Typically within our industry, there's a range of host cell types that are used. So, for example, we would typically use the E. coli or the Pichia technology. Within mammalian, we would typically use engineered Chinese hamster ovary cell lines. However, in the industry, there's a variety of expression technologies that are used within that platform. There's the NS0 platform as well that was used in the past, which was based on mouse melanoma technology. But then, yes, you are seeing more trends coming up with plant-based technology, mRNAs coming up as well. So I think now we're getting more diverse spread across the industry of different host cell types that are used.
Which cell line is the most widely used?
Within mammalian, I would say absolutely the Chinese hamster ovary is the predominant workhorse that we would typically use in the industry.
I guess one of the main requirements is like the speed of growth of these cells, since you want them to produce a lot.
Exactly.
Is this the reason why human tissues are not used so much, or is this also a question of ethics?
I think it's both. So ethics absolutely has a part to play. However, when you look at the economics of a process, to be able to use human cell lines is just not as economical than it would be to be able to use suspension variants of a Chinese hamster ovary cell line.
Quite a pragmatic answer. Yeah. Okay.
But saying that though, over the last 30 years, I would say the Chinese hamster ovary host cell lines evolved so much in the sense that there's a lot more genetic engineering elements that have come into play. And the genome's been fully sequenced, so it's fully understood. So we know exactly what genetic elements to be able to take out and to be able to replace to get us the optimal workhorse and system in the bioprocess field to be able to express that product at the maximum amount that that cell line could produce.
Was it a serendipity that the Chinese hamster ovary cell, so CHO cell, was chosen as the ultimate workhorse for mammalian manufacturing, or is this just a historic development?
I think it was a historic development element to it. There was a big breakthrough, I guess, back when we were students, still 30 odd years ago, where the Chinese hamster ovary cell line was matured and developed in a way that we could, one, for example, engineer it so it could produce molecules with the sufficient glycan profiles like a human cell line could. And from there on out, I think it just took in the industry. But as I said, most of our industry would, for mammalian modality, would use the Chinese hamster ovary.
Ronnie, for bi, tri, multi-specific antibodies, do you always work with CHO cells, or is there space for some diversification?
Yeah, CHO cell is the go-to cell line for us for multi-specific antibodies. Of course, in research stage, we oftentimes use HEK cells as well because it's pretty robust and quick and has also the necessary post-translational modifications, but downstream still mostly CHO cells. And like Atul said, the technology advancement has really been remarkable. We've seen multi-specific antibodies achieving grams per liter productivity in the downstream process is really remarkable. They can be produced like labs.
Atul, how do you make decisions when selecting cell lines?
So I think it comes down to three elements really, Martina. So for example, if you're in discovery phase, like Ronnie said, mostly the industry would use HEK 293 cell lines because you want the readout really fast. And it's good enough material to give you a sense for, how efficacious or how that molecule is going to perform.
For like the early toxicity
for
assessments,
very,
right?
very early discovery phase. So the driver behind the selection comes from that. The second part of it is the technical element. So do you need to have post-translation modification or do you not? So therefore you'd go either the Chinese hamster ovary or the E. coli route. And then the third element is the economics.
And when it comes to scaling up, is the only thing that changes just the size of the bioreactor? And I've seen a 20,000 liter reactor. That thing is enormous. But do you have to consider anything else here?
Yeah, there's a whole array of parameters that you need to think about. The vessel design, for example. So I alluded to, in clinical, people would typically use single-use technology now for single-use bioreactors for the production of first-in-human batches. You then have to switch to a stainless steel-based technology because 20,000 liters, like you saw, they're big vessels. Single use isn't an option, and therefore you have to switch between the material type. With that also, you have to consider the method of mixing, so the spargers that you use, the impeller design that you would use, the gassing strategy that you would use, because some vessels are taller and bigger, wider, and some vessels are thinner and longer. So you've got a number of different parameters, various geometrical issues that you'd have with the vessels, process parameters such as the pH of the cell culture, the dissolved oxygen tension, etc., the bubble size. There's a whole podcast around scale up, Martina.
Oh,
absolutely. And also, I just alluded there around the cell culture process. However, when you come to primary recovery, where you're trying to separate the cell material, cell debris from the product, there's different technologies as you go scaling up from filter-based technology to centrifuge-based, and purification.
So I would like to come back to what you said at the very beginning, the new molecular formats, right? How does the manufacturing differ for them if we go a bit more complex?
Really good question. It starts with the expression area and the screening tools that we use around the expression technologies. However, what we screen for, the number of clones we screen, what we're looking for would vary. So we, for example, might screen more clones. The most diversity would be in the purification aspects. So each of these type of molecules would come with it a uniqueness around what you need to purify out and keep.
Interesting. Ronnie, is this maybe a consideration also for early discovery? Do you need to keep in mind the purification stage when designing the structure for your bispecific, trispecific, or multispecific products?
Yes, absolutely. Because, even in early stage, maybe the requirement for purity is not as stringent as downstream manufacturing, but we also want to get it as pure as possible so that our biological assay readouts are reliable. So we really have a lot of thoughts put into how to design a molecule to reduce, for example, mispairing.
When it comes to pushing the boundaries of this area of research and manufacturing, how does innovation look like on a large scale when you're already at the maybe even commercial stage?
So I think when we come to innovation in large scale and commercial, it's really focused around improving process robustness and ensuring that we have a lower cost of goods. A lot of the innovation on large scale is really focused around the drivers behind what you need to achieve that scale, which is typically around process robustness and ensuring the best-in-class quality.
Mm-hmm.
So process robustness would come down to, for example, implementing in more inline testing technologies, so PAT tools, introducing more automation, for example, to tighten the control that you have of the process.
Uh-huh.
Another part of the innovation when it comes down to the commercial activities is now around reducing cost of goods for our customers. And what you'd hear more and more lately in the news is around process intensification, for example. So there's a switch between what we would term in the industry fed batch, traditional fed batch processes, into more intensified processes. So what you're trying to do now within a shortened period of time, trying to get more product out of that bioreactor and purify the material much quicker.
When you say fed batch versus continuous, does this mean that instead of giving your cells a meal at the very beginning, you keep giving them more food as they grow?
Yeah, so it depends on, there's a variety of different flavors of continuous, and the one I was alluding to really was process intensification. And using your analogy, it's having a bigger meal to start with, more food on your plate to start, and then trying to eat it as quick as you can.
Right. Sounds like Thanksgiving dinner, something like that.
Absolutely.
Okay. I'm loving this discussion. We have covered the discovery and manufacturing processes at various scales needed to produce antibody-based therapeutic products, including selecting optimal cell lines and using advanced purification techniques to ensure high titers and purity. Atul and Ronnie have also discussed what innovations are being implemented to push the process efficiency and speed even further. Now I would like to maybe look a little bit forward when it comes to space of antibodies and multispecifics. Ronnie, what are you looking forward to in the next 5 or 10 years in this space?
Yeah, I'm looking forward to and also hoping to see quantitative leaps in improving solid tumor treatment, because I think that's still a huge challenge in the field. But I think we are actually now seeing more approved or about to be approved antibody-based or bispecific antibody-based medicines for solid tumors. I'm hoping to see more of those in the next 5 to 10 years. And also, again, more advance in digital biology tools to help us to design and make antibodies better and faster.
That sounds great. What about you, Atul? What excites you most about this field when it comes to manufacturing antibodies?
I'd like to build on what Ronnie was saying, actually. So what really excites me and what I'm seeing more and more in the industry being reported on is around precision medicine, more targeted therapies, and improvements in delivery systems. For example, the blood-brain barrier, there's a huge amount of innovation that's happening around that space. I think we're just enormously privileged to be in this industry. Just being looking at the diversity of modalities that we're seeing the advancements that are coming through when it comes to discovery phase, development, manufacturing, how we can actually lift technologies from other industries, from the tech space to be like generative AI. How can we use that to disrupt our industry? It's a fascinating field that we're in, and I for sure am really humbled to be in this industry.
Fantastic. Well, thank you both for your time. It was a great discussion, and I hope our listeners also enjoyed it as much as I did.
Thank you.
Yeah, thank you for including me in this fascinating conversation. I learned a lot too.
That's great. What a great discussion that was. While the biologics pipeline is still dominated by monoclonal antibodies, we are increasingly seeing more and more focus landing on bi, tri, or multi-specific antibodies. These proteins can bind to 2 and more antigens, leading to synergistic therapeutic efficacy, and potentially enhanced therapeutic efficacy and reduced treatment complexity. It is safe to say that these molecules are transforming medicine and making an impact on patient lives. That's it for today's episode of A View On. Thanks for joining as we explore the future of pharma. Make sure to subscribe to the podcast and follow us for more episodes diving deep into the industry's latest trends.